All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. We discussed messenger RNA present in human breast milk in a study, I think about 5 days ago. What that study said was that there are trace amounts in picograms, 1 to 2 picogram per milliliter, of the messenger RNA from the vaccine present in the human breast milk. And I had noted that even if it is trace amount in the milk, of course, there has to be a higher quantity in the cell, because whatever is in the cell will not all become extracellular vesicles. And that also means that there will be even higher amount in the circulation, because everything in the circulation will not end up in the breast tissue, and that will not end up in the extracellular vesicle. So there were many questions about other tissue barriers and the possibility of mRNA being there. There were questions about this mRNA going to the child and what will happen. So we did a Zoom call and we discussed some of that. And I thought it will be reasonable to discuss some more barriers. That also means that what I'm going to discuss is not the study itself. The study only talked about the blood milk barrier, or the vaccine having crossed that barrier and mRNA appearing in the milk. Study did not talk about the blood testis barrier or blood thymus barrier, thymus, or blood brain barrier, or um, other tissue barriers. However, I wanted to have a look at other tissues today and see what are the similarities and differences of these barriers and what are the possibilities. So, this information that we will discuss today is not from these studies. So, keep that in mind. So, it is more of a mechanism discussion than a study discussion. So, let's start our discussion. So, first of all, this is this is the plug for Dr. Bean. So this is drbeen.com. In the description of this video, there is a link for drbeen.com, and the price is under $100. And I think you would love it. There are more than 900 lectures here. And these lectures, some of these are not on YouTube, and these are fun lectures. So see if you want to take advantage of that one-time price. So here is the first opening article that we look at. This is the structure and function of blood tissue barriers. All of excuse me, all of these links are present in the description of this video. Structure and function of blood tissue barriers. So generally, the function of blood tissue barrier is to prevent proteins and macromolecules and cells. Macromolecules are bigger molecules, proteins, cells, from getting out of the blood vessels and entering the tissue. So cells are generally not allowed to get out, except the immune system cells that have specialized ways to get out. But cells like RBCs are not allowed to go out. Similarly, proteins are generally not allowed to go out. There are tiny proteins that can go out and be used, or amino acids can go out and come back. But for example, albumin or globulins, these are not allowed to get out. And if they do, then we develop edema and such pathologies. However, in the case of blood tissue barriers, there is even more strict mechanisms more hard mechanisms, more barriers, more isolation, more restriction from anything to go from the blood towards the tissue or from the tissue back towards the blood than just those things that are specialized and specially allowed. So normally what happens is, I'm going to draw some of these parts. So these are our gifts for humanity and they're continuing. So normally what happens is, imagine this is a blood vessel. The internal surface of the blood vessel is covered by cells, heart and blood vessels. These cells that are facing inwards are called endothelial cells. Endothelial cells. Then usually outside of the blood vessel, there are various layers and basement membranes. Some blood vessels have muscles in them as well, and so on. Depends upon the kind of blood vessel, how thick it is, and what tissue it is going to. And then outside of the blood vessels are the tissue cells, right? The barriers are formed in multiple ways. For example, in the case of blood-brain barrier, the blood vessel wall has tight junctions between the cells. What does that mean? Normally, the blood vessel cells, especially in the capillaries or smaller blood vessels, especially capillaries, the blood cell or endothelial cells are put together in a way next to each other that there are gaps between them. Through these gaps, substances can get out and come back. So imagine this is inside of this blood vessel and this is outside. Usually, outside of these cells, endothelial cells, there is a basement membrane. So even when there are gaps here, through which some substances can get out and come back, still larger particles, for example, antibodies, or for example, albumins or globulins, they cannot get out because of this basement membrane. So you might think that, all right, then how do the blood cells, for example, neutrophils or monocytes or other immune cells, how do they get out? They actually they drill holes in this basement membrane. And from here, they get out through those holes and go to the tissue and do their function. Now, in the blood-brain barrier, for example, this is not allowed at all. So what happens is, for example, in the blood-brain barrier, the cells have tight junctions between them. So as I was mentioning, that cells usually have slight gap between them. But if these cells are tightly fused with each other, they are individual cells, but they still are so closely pressed with each other that there is really no space between them for movement of substances. And there are specialized protein structures that are called tight junctions, and these would help seal these cells when they're pressed with each other. So in the blood-brain barrier, for example, there are tight junctions, and those tight junctions prevent blood or the particles from the blood to get out or things to get in. Then in the blood-brain barrier, there are even more cells wrapped around the blood vessels. So there is further fortification of the cells. Pericytes are present. Then more cells are present. Astrocytes are present. Their foot processes are present. The point is, it is like 3-cell thick blood-brain barrier. So then let's say this is all brain tissue and this is blood. How do things then get out? So normally, some things can move. For example, water can move. But other than that, usually the brain tissue cell and these specialized cells that are wrapping this barrier. They have specialized channels in them, they have vehicles in them. And if something needs to go from the blood to the brain, it will have to become attached to that channel. The channel would then decide to bring it to the other side. So, point of this all is, one, barriers are fortified structures. Secondly, the tissue on the other side that these barriers are protecting still needs substances to work with, to use, to utilize. And for that, there have to be specialized channels that help move these substances. This is in contrast to common tissue capillaries, where there is just very simple gaps between the cells through which substances can move. All right, so now if I go back here, the we will start from the blood milk barrier and how does mRNA end up in the milk. This mechanism is actually not discussed in that study other than a conjecture. So I hope that you have actually gone through that study because we discussed it. So I'm going to not repeat that. Instead, let's see what happens. Imagine this is a blood vessel. The reason for me to do this is then I'm going to extrapolate that to the other barriers. So let's say this is a blood vessel. It has endothelium on the inner side. And let's say this is the breast tissue. Then here in the breast tissue, my marker doesn't respond very well. In the breast tissue, there are cells whose job is to make milk. So let's say this is that structure. The milk will be produced by the cells here. And these cells will produce milk. That milk will then be exited in these little tubules. And from there, all of these tubules are eventually called lactiferous tubules. Eventually, many of them will come in and open at the nipple. This is general high level structure. The question is that this cell over here, this cell, for example, how does this take things from here, from the blood, and bring them to the milk side? How is milk formed? So remember, breast tissue is a modified sweat gland. So what happens is, I'm going to make one cell. Let's say this is a breast tissue milk making cell. All of these cells that do secretions, they usually are called to have two sides. They are called to have basal side or a base and an apical side or an apex. Basal side is usually the one that is resting towards the tissue. So these sides will be called basal sides. And then apical side is the one that is usually the conical or narrow part of the cell, and that usually faces the area where secretion of the cell will go. Generally, these cells, if their secretions are going to go to the surface of the body, for example, sweat gland or the mammary gland or the glands present in the anorectal area or the glands present in vagina or the glands or the surfaces present on the eyes and mouth and nose and GIT, their their secretions go to the surface. And such cells are called epithelial cells. So this is an epithelial cell with a basal side and an apical side. And let's say this is the mammary gland tissue. So now what happens is, from the basal side, the blood vessel that is under or behind it, there are specialized channels through which the cell selectively picks up things. So then the first question will become, okay, fine, how will messenger RNA enter it? And the answer is that messenger RNA is wrapped in a liposome, right? So lipid nanoparticle is a liposome. Liposome are structures that are made up of lipid. You may have actually seen many supplements, many drugs or herbs are wrapped in lipids. Now, drugs are wrapped in lipids as well. For example, let's say for breast cancer, you want a drug that would go to the milk tissue of the breast. Now, that tissue is separated because of the barrier. So, what you do is you wrap that drug in a liposome so that it can cross the blood tissue or blood milk barrier. It is a standard practice to wrap the molecules in a lipid particle so that they can cross the barriers. And you can actually Google it. You can say lipofication or liposomification of a drug to help cross the barriers, and you'll come across thousands of articles and studies. So here, mRNA is wrapped in this LNP. And because of this LNP structure, lipid structure, this mRNA and LNP they do not need any channel. They would simply come in. They do not need any help. They will just come in. So the problem is not that they just came in. The problem is they were in the blood. Because they were in the blood, they did cross the barrier. And crossing the barrier is not odd. They will cross barriers because they are lipid wrapped structures. Once the LNP is inside the cell, then inside the cell, the mRNA is going to become released, just like it will be released when you inject it in the muscle, and the muscle cell will pick it up and the mRNA is released. That means even spike proteins will be formed. Now, all of this discussion that I'm doing, that study did not talk about this. So it is not the study. It is me providing a mechanism which I could be totally incorrect. But if I am incorrect here, then the same mechanism in the muscle can also not occur. So if I am wrong, then muscle mechanism will have to be wrong. And if muscle mechanism is correct, then my mechanism here will have to be correct as well. So as the mRNA is released, that mRNA is going to give rise to making spike proteins. Those spike proteins will meet the same fate or do the same thing as they were going to do in the muscle as well. Will this cell then present parts of the spike protein on its surface? Yes. Will some spike proteins will come out? We have seen now from various studies. Yes. So that study actually did not look at the spike protein. So again, I keep clarifying, these mechanisms are not attributable to the study. They did not look at those things. So I hope this is sufficient for me to say it once or twice instead of having to repeat throughout the discussion. Now, this mRNA, when it is present in the cell, and somebody asked me this question, that will this mRNA be more in quantity than the virus's own mRNA? If the virus SARS-CoV-2 lands in our body, that's also going to generate more viruses and more mRNAs, and they might actually reach these places too. Will they be more or this be more? So look, vaccines mRNA quantity is at most the amount that you have injected. This discussion that mRNA will start becoming integrated and then becoming replicated, there is no proof for that yet. Excuse me. So the mRNA that is present is here, some of that, a fraction. Now, here is the delicate part. When this mRNA and some spike proteins and the other proteins in the cells and whatever is in the cytoplasm. Cytoplasm is the fluid and the things outside of the nucleus but within the cell. Check this out. What happens is a part of the cell breaks off. This part of the cell breaks off. So let me see if I can actually just erase this much to show you an animated break off. So now this part has broken off. That part that has broken off is the extracellular vesicle. Extracellular vesicle, and this is milk. So when the milk is formed, I mean, not this, the whole milk is not this, then this would get ruptured, some things will come out, there are specialized secretions in here as well. But this is how milk is formed. That means cells content in the cell's cytoplasmic contents are part of the milk. That is how milk is formed. So this is one way of creating a secretion. And I would then show you that this, what I'm saying, is totally correct. So let me go to, here is the mammary gland. And again, if you said that, hey, Wikipedia is kind of not trustable, you can pick up any physiology book and histology book and look for the same terms over there, mammary gland, and then you can see the structures there as well and functions. So if you see here, a mammary gland is a specific type of apocrine gland. Now, what is an apocrine gland? Here is the apocrine gland. So if you see here, this is CFCF, their own work. If you see, this is a cell made some milk related stuff, and then a part of the cell broke off. So pinched off portion of the cell is the secretion. So, and if you see here, this is an apocrine gland. That is how the milk is formed. So that means if there is mRNA inside the cell, that will be in the milk. If there is spike protein inside the cell, that will be in the milk as well. If there is some other things, the milk itself, that would be in the milk too. If there are other parts of the cytoplasm, they will be in there too. So that is one important understanding. On the Zoom call, I had done this discussion that it is not necessary that whatever is present in the cell, all of it will get out in the milk. Actually, when the extracellular vesicles are formed, there are other vesicles that are formed too that can actually go in the other directions. This big vesicle is going to go to the milk, but smaller, tinier extracellular vesicles emerge from the cells as well. That is one. Number two, the amount of RNA in the cell, all of it is not going to become milk or part of the milk. Some of this is just going to stay here. A fraction is going to get out here. You can think about it this way, that everything that is in our blood will not become milk. A very tiny fraction. If a woman, let's say, drinks 3 liters of water, that 3 liter would all not become milk. So a fraction. That means that whatever is in the blood is going to be in higher quantities compared to what came out in the milk. So the thought that there is trace amount is interesting, but that is an indicator of greater amounts behind this one. Now we are done with the milk discussion. The second concept I want to discuss is sweat. Do we think there can be mRNA in the sweat? And again, there is no study. This is not a study-based data. This is a mechanism. Now, sweat is actually, mammary gland is a modified sweat gland. So if I go here for a sweat gland, the sweat gland actually has a very similar structure. So I'm going to talk about that in a second. Here is the diagram of a sweat gland. And in the sweat gland, there are various types of gland tissue cells, but there is a barrier here as well. If there was no barrier, then we would just start leaking stuff that is inside our blood in the sweat. And that would be a problem. So here the question is, will mRNA come out in the sweat? And this is a conjecture now, based on this. And here, imagine this is a sweat gland. Sweat gland is different. Mammary gland makes milk, sweat gland makes sweat. But the structure, functional structures are very similar. There is a blood vessel. Inside the blood vessel, there are substances that are of use to make sweat, including water and sodium and potassium and so on. There are specialized channels that will pick up things from the circulation that are necessary for the sweat glands to make sweat, And then the sweat gland will make sweat and release that into small canaliculi or small tubules. There, the sweat is then concentrated, and there are more things that happen to it. So then the question becomes, can sweat have mRNA? So we're talking mechanism. If this LNP present in the circulation, only then the mRNA would enter a mammary gland cell. So let's have this very important, delicate point exposed. This will not happen that mRNA alone, naked mRNA without lipid, somehow enters the breast tissue, milk making cells. It will have to be wrapped in a lipid nanoparticle to be able to enter the tissue, the cells. Otherwise, it cannot. Same is going to be the case for sweat as well. So if we hypothesize that mRNA wrapped in lipid nanoparticle is circulating, then that mRNA particle in the particle will end up in the sweat gland tissue cells as well. And if it does end up, it is going to do the same thing. It's going to come out, and then some spike proteins will be formed, and then the sweat will be formed. So if I go here, I'm trying to see what is the best place. This is mammary gland, sorry. Apocrine glands, sweat glands. So here, these are various sweat gland. They are acrine and apocrine and apoacrine and so on, various kinds. At the end of the day, there is going to be sweat that will be produced, and that will go into the lumen. Lumen is that little canaliculi in which you put that, and then that canaliculi opens on the surface. So let's say this is skin. On the surface of the skin, a small tube will come, microscopic tube will come and open here. And that tube is then directed or continuous, and the surface of this tube are the sweat gland cells, and they'll make sweat, and then the sweat would appear here on the surface. So we are talking about this cell over here, and this looks like a penis, my apologies. So this cell over here, if it is a sweat gland, then LNP would go in there, mRNA will be there, even spike proteins could be formed. They will be destroyed in here by the endosomes, by the microautophagy, many other mechanisms. Some of this will be presented, some of this would spill out, and some of this mRNA and the spike will end up on the sweat as well. Would that do? So because some folks challenged me that, hey, you are somehow talking about the vaccine and so bad, why don't you talk about virus as well? So yes, if there is virus infection, then these can happen too. The only condition is the following. The virus, so let's say that we want the viral mRNA to come out in sweat. For that, here is, let's say, sweat gland cell. For this, we want the virus's mRNA first to be present in the blood circulation, right? And the answer is, yeah, it can be present in the blood circulation. It can be present in the tissue. There is an infection. The viruses are forming more viruses, and they're making more mRNAs. And we have factories of our tissue making viruses and mRNAs. And lots of those cells are breaking, and the mRNAs and the viruses are just, and other enzymes of the cell, they are spilling out. Local inflammation is occurring that would cause inflammation of the blood vessels, and the blood vessels will become permeable, and these things will enter the blood circulation as well. Yes. Now the question is, for this mRNA that is now running around, let's say, because of the virus in our blood vessel, for this mRNA to end up in sweat or in milk, the mRNA has to cross this barrier and enter the cell. In the case of vaccine, the help is that we provided an LNP packaging. That packaging acted as a vehicle to help the mRNA cross this membrane or barrier. In case of the virus or the RNA is not packaged in a lipid nanoparticle. Instead, the RNA is actually packaged in the virus. Virus has to do a specific way action to enter a cell. It has to bind with ACE2, then it has to fuse, and then the RNA would enter. Now, if just the RNA is running around, then that will not enter a cell. Even if that is RNA coming from the vaccine only. Imagine in the muscle tissue, the RNA got exposed or removed, the LNP got removed, and the naked RNA got spilled. Even if that goes in the blood circulation, it can't just willy-nilly enter cells because RNA is charged and it needs packaging to enter the cell. That is the same thing for the virus. So then you can say, what would happen? The virus is accompanying that too. Fine. If the virus is also here, then virus will have to infect a gland, mammary gland or a sweat gland or any other gland, to be able to bring the mRNA here and then to be able to have those viruses and mRNAs in the sweat. What does that mean? That means we'll have to find out ways for the virus to infect the skin. We haven't yet seen this virus infecting the skin. Why? Because there may not be enough ACE2s or the access to the virus to the skin cells, just like it can have access to the pulmonary tissue. But to enter the skin, the viral RNA will have to be packaged in a lipid particle, or there would have to be enough destruction in the skin to allow the virus and stuff to just spill in the cell. But if it does spill in those cells are destroyed cells, they're infected cells, I don't know how much they would function, but that simply means this destroyed tissue will just let whatever comes to it outside, and that is bleeding on the skin. So can virus RNA appear in the sweat in the same way as the vaccine RNA? No. Why? Because vaccine has lipid RNA particle on it. It has been lipidified, while the virus will have to come himself or herself or itself to have the RNA enter a cell. So I hope that clarifies that part. So if the sweat gland, can it have mRNA? Yes. If mammary gland can have it, then sweat gland can have it too. How much? I don't know. Is there a study? No. Or at least I haven't seen it. Maybe there is. Can spike proteins be there? Yes. Did this milk study show spike proteins? No. Did they look for it? No. But if there is mRNA, now some folks made this comment. They said, well, maybe the RNA that entered the cell was not viable RNA. The authors of the study said, we cannot tell you, this is a limitation of the study, if the RNA that is released in the milk was culturally viable or not, meaning could it make spike proteins or not in the baby. Now that is a very different question and answer compared to this side. We are talking this side. So let's say over here is a baby, and the baby is going to drink this milk. So they are talking about the RNA pieces here going to the baby. The question is RNA here going to the cell. So let's think about it for a second. If we say that RNA entering the cell is not viable, then that will mean what we are saying is that RNA is wrapped in the lipid nanoparticle because otherwise it cannot enter, and it is damaged. That is kind of a problem. Why? Because the lipid nanoparticles from the vaccine have viable RNA in them. Maybe some of them have damaged RNA as part of production, but generally they have viable RNA. And RNA cannot enter the cell unless it is packaged in a nanoparticle. And if it is packaged in a nanoparticle, then that is a viable RNA. This is like if I send you something which may be delicate, but I have a fortified metallic box around that. And now I say that, you know what? Although this thing entered here in the cell with the nanoparticle, with the metallic box on it, but I think that the stuff inside was broken. That is hard to believe. That means that mRNA that entered the cell is most probably viable. However, the one that exited the cell in the milk may have been broken because as it is inside the cell is going to start working on the mRNA, make spike proteins, and chop up the mRNA into smaller pieces. And that would cause the mRNA to become less viable. So, sure, I think so. This is me thinking. That means totally incorrect. I'm talking mechanisms. If anybody want to challenge this mechanism, I'm totally happy to see which book they want to talk about. If the mRNA is getting in the milk or sweat, then some of that is going to be viable, some of that is going to be not viable. Then we should answer this question here as well, that is that really dangerous for the baby or not? They said, I don't think they commented on that. So again, I cannot say anything from a studies point of view, but they only said there should be a concern for at least 2 days after the vaccine. And then the Journal of Pediatrics, I think they came out and after this vaccine and they said, after this study, and they said that women should not or be careful while feeding babies at least 2 days after the vaccination. So there seems to be a concern. I would just live with that. I have no idea. We are, I'm not a neonatologist or a pediatrician to say when a baby is given a tiny amount of a protein which can be an mRNA like structure, not a protein but nuclear material, what would happen, I don't know. But we eat meat all the time, some of us, and that meat contains cells, and those cells contain DNA material, and we digest it, break it down, and kind of don't even feel it. I do not know about the baby, and neither the authors nor the journal try to make a conjecture, so I will also not make a conjecture. Okay, so sweat discussion done, mammary gland discussion done. Now the third is testis. So question to you, we are already 38 minutes into it. Should we stop here and discuss testis and brain at another time, or should we keep going? More coffees for Dr. Bean, or Rima says our body handles proteins all the time. Yeah, we eat proteins all the time, so we don't have a problem with that. Even uh, DNA or nuclear material all the time, we eat that as well. Okay, so keep going. So let's keep going. Please hold your questions. I want to finish this discussion of the barriers and then see, maybe we can do a chit chat afterwards. Okay, so now we go to the next barrier. We have done sweat, mammary gland. I'm going to just close these. That is done. Sweats is done. Mammary gland is done. Now, let's talk about the blood testis barrier. This is the most interesting one. So, blood testis barrier is, I think it is very interesting. So let's talk about it. What happens is all glands have this thing that there is a tube that opens up on the surface. The back of the tube has a factory to make whatever secretion. In this case, the tube do open up on the surface, but they are not making just secretions. Of course, prostate will make secretions, but there are sperms as well. Of course, the testis would also make secretions, so the sperms can, float in them or swim in them. If we make sperms that are in the dry area and they do not have anything to swim in, then how would they travel? So there are secretions as well, plus sperms. So what happens is the following. Imagine This is a, I'm going to make a couple of cells. So let's say this is a cell. These cells are called Sertoli cells. And I'm making a very specialized structure. I hope I can successfully make it. You like it, I think. Can you see this little fin-like structure? This, just like a fish has a fin. So this fin-like structure, and imagine if I put one more cell next to it, if I make a copy of this to speed up this process, layer panel open like this, and then bring them close to each other. Now, here you are seeing two cells next to each other. And what happened was their fins kind of join hands. And similarly, if we have one more cell, let's make one more cell just for the kicks of it. So there is one more cell, and that is connected here. And there, this cell's fin is fusing with this cell. So let's say this is cell A and B and C. Their fins are kind of, they're holding hands. But here is the fun thing. There is one, because of this little barrier they are making, these cells. And imagine if I make it smaller, imagine these cells are sitting in a rounded structure. Right? Like this. And all of them are holding hands with each other. Right? So they all have these fins that are fused with each other. Can I now say that this structure has two parts to it? Although these are one single cell, these are Sertoli cells. This is a cell, for example, let's just focus on this cell A for a second. This is one cell, but can I say that this part of the cell is exposed to a different environment versus this part of the cell, which is exposed to a different environment? Because they have these fin-like structures. And so the fluids here cannot cross this to go onto this side. And the fluids and substances here cannot cross this fin-like barrier to go into this side. This is blood testis barrier. Blood is on this side. Things are coming out and they're providing nutrition and everything. And the fluids and sperms that are going to go out, they are on this side. So this thing, this structure is then continuous in a tubule, and finally that tubule would open up with the prostate and finally urethra on the surface. So this side here, this is such a beautiful mechanism. I love mechanisms for this reason, and structures and everything. So here, this all is the sperms plus the fluids and substances, the nutrition for the fluid, the environment maintenance. There are even proteins that would help the help. So the sperms go with all lots of things with them. So they have proteins with them that would reduce the woman's immune system cells activity, so that those cells do not accidentally kill these sperms. So sperms have a lot of paraphernalia going with them, but that all is here. Some is going to be picked up from prostate and so on. This is a privileged area. What is it privileged from? This is privileged from or protected from this area. This is the blood area. Blood vessel has the blood, then these are the interstitial fluids. These fluid can actually bathe this whole thing up to these fins, but not further inside. And inside here is the fluids of the of the secretions that are going to take the sperms out. So you can ask this question, that where the heck are the sperms? So sperms are actually produced by cells that live here, called spermatogonia. These spermatogonia, little cell, or the, you can say in rough terms, they are the um, stem cells for the sperms. So they divide by mitosis and make sperm cells, or um, spermatocytes. These cells, the tiny cells that take after division, they are formed. They start going through these Sertoli cells all the way towards here, and by this time they develop a tail, and they develop a head and the structure of a sperm, and then they are released here to say, all right, man, go do something, right? So then the question for which we are here: can mRNA be in here? So let's look at this structure together. mRNA and lipid particle is here. This cell, I'm going to delete this little blue thingy. This cell, spermatogonia, is actually exposed to the mRNA and lipid nanoparticle. And just like Sertoli cells and other cells, it can enter here. Is this a study? No. Is this a mechanism? Yes. Mechanism that the spermatogonia are exposed to the blood side. But once they become spermatocyte, or they give rise to spermatocyte, and this spermatocyte start going outwards and they cross this barrier, interestingly, then the spermatocytes are not exposed to this side anymore. So could there be LNP here? In theory, it can be, because this little barrier is made up of lipids as well, and lipid nanoparticles can cross lipids. So in theory, yes, it can be there. And then it can actually be in the sperms as well. But there is no study that shows it. In fact, I wanted to show you this study. As you can tell, I've been reading today. Sperm parameters before and after COVID-19 mRNA vaccination. This is a study from June 17, 2021. And the author affiliations are the Department of Urology, University of Miami, Florida. So a good university, a good set of research. So good research. And here they say, for the Miami University, they recruited boys from the university, youngsters, to say that, hey, can you give us semen before and after the vaccination? And this is a very interesting study, because the semen's quality and the sperm quality actually had improved after vaccination. So let's read it for a second. I still want to go back to that. So if you see here, semen analysis was done: semen volume, sperm concentration, sperm motility, and total motile sperm count. That is what they were counting. 45 men, so small study. Half of them, almost 46.7%, had Pfizer BioNTech, 53.3% had Moderna. So after the second vaccine dose, so they collected the semen before, and then I believe, how many days? I believe 70 some days after the second dose. Baseline samples were obtained after a median abstinence period of 2.8 days, and follow-up samples after median abstinence of 3 days. Of the 45 men, blah, there was a 75 days, median of 75 days after the second dose. Okay, so after the second vaccine dose, the median sperm concentration significantly increased to 30 million per milliliter, and this increased to that from before this, it was 26 million per milliliter. And there is actually a possibility that because they were abstaining and doing it, so it is possible that it was increased. So it's not that the vaccine went in and said, all right, more sperms will be made. Okay, so, and the median TMSC motility to 44. So TMSC was the total motile sperm count. So total motile sperm count, that went up from 244 million, and this was 36 million. So again, kind of not significant either way, but it didn't decrease, it actually increased. So, and these are significant data results. So at least the counts were there and in the correct amount or even more. The functioning of them, their motility was fine as well. So at least there is a study that shows that much. Now, if I go back here, the lipid nanoparticle, if present here, can it cross this? Yes. Can it cross here? Yes. Can it just go and infect? I'm calling it infect. Enter the spermatogonia and then become part of the, as a spermatogonia divide, can it become part of spermatocyte? Yes. So let's look at that as well. So here, these little blue things are spermatogonia. Do you see these red ones are Sertoli cells, or the supporting cell? They are the mother cells for the sperms. And if you see here, do you see them kind of shaking hands with each other and binding with each other? This is the blood testis barrier. And it is interesting, blood is on this side, the lumen where the sperm will be formed and then get out is on this side, and here this one cell lives in two eras. On this side, on the basal side, it is exposed to the fluids, blood fluids, and spermatogonia are exposed to that as well. And then on the other side, the same cell is now exposed only towards the seminal fluid side, and the spermatocytes are here, which are also not exposed to blood anymore. So if their parent spermatogonia had something in it, when the parent divides, these will have that in it as well. Now these cells will start losing cytoplasm. So that is the other thing to keep in mind. If there is mRNA present in the cytoplasm, the sperm kind of sheds the cytoplasm. It leaks the cytoplasm out and just kind of have mostly the DNA material in it. It kind of sheds the rest. It does keep some nutrition with it because it doesn't want to go hungry when it is traveling all the way from the penis to the vagina to uterus and uterus and fallopian tubes all the way to ovary. It needs something for the road. So it does keep some nutritional substances with it, but majority of cytoplasm is exited or dried up. So will the mRNA stay viable here or lipid nanoparticle? I don't think so. But again, there is no study for this. So this is the blood testis barrier. Now, we're talking within the sperm cell. Could there be mRNA and LNP just leaking out because they can go into the cell? They can go into the cell. The cells could produce spike proteins and do the same thing as other spike proteins do. So this is done. Now the last part, there are three more barriers. Blood thymus barrier, blood retina barrier. There are two barriers in the retina, internal and external. So blood retina barriers, eyes, and then blood brain barrier. So I'm going to not do them. This already 1 hour. Nobody's going to watch this beautiful thing that I have generated for you all. But this is the discussion for the possibility of these little LNPs to be present. If they are present, Alicia is laughing. Alicia, nobody watches these long videos and then they sit out there saying, please watch, I have some treasure in me. So if LNP containing mRNA is present in the blood, then the rest can happen as well. Now, there have been previous studies that did not look at LNP with mRNA. And please don't bring up the Japanese study that was not looking at both of the things. It was only looking at LNP piece. This milk study is the first ever study that actually conjectures. It just says that, hey, in the milk there is mRNA. And now from the muscle to the milk, there is a mRNA. What happened? Of course, mRNA reached here for some reason. Somebody said that, well, this must have been contamination. Sure. How was the milk contaminated with LNPs? Or how I don't know, maybe the woman had some vaccine that she just massaged on herself and then that contaminated the milk, or as they collected the milk they put some vaccine, I don't know. But if there is mRNA in the milk, then it had to go all the way from here through the circulation. And this, the conjecture in the study was either through the lymphatics, the LNPs enter the blood circulation, or directly through the permeable blood vessels enter the blood circulation. From there it was roaming around and it went to every tissue. This is not what they said. And then it went to breast tissue as well. So if that is the case, then all these other possibilities are possible as well. So now, would you like to have the same discussion in this talk, or should we do a chit chat and have the discussion, or good enough 1 hour done with Dr. Bean with weird diagrams today? You decide. So John Snyder says the real question, does mRNA in the milk end up at the other end of the baby? I think it depends upon the baby's age and how mature their GIT is. Will they be able to break it down? It's a nuclear material. They should be able to break it down. That's not going to be a problem. Okay, so let's see. Chit chats. So chit chat will be questions around this discussion. Chit chat separately. Okay. All right. So I'm going to hang up. Please do me a favor. As you can tell, I take a lot of pain to put these beauties together, the lectures, long lectures. Share them, subscribe, like, comment. When you comment, the YouTube algorithm thinks, oh man, this is something really important and interesting. I should show it to others too. And the best is to support this work. There are links in the description. You can buy Dr. Bean, 900 more videos, me doing such things. Or you can use PayPal, or you can use Buy Me a Coffee, or you can become part of patrons. Interestingly, I saw today 7 patrons left. So instead of more patrons coming in, there are patrons going. So anyways, you become patron, or you can even buy YouTube's membership as well. And at the end of the day, it's not necessary. These are gifts for you. So thank you very much. I'll see you in a few minutes and we'll do some chit chat. Let's see if we can keep our questions to this topic.